Well, also I would like to thank the organizers of the meeting, in particular Roger, Tomer, Michal, and Dana. So it's a great pleasure to be here. You heard that I'm both German and American, and I'm actually also Jewish. So as you may know, the Americans have the reputation that they always start their talk with a joke, and the Germans have a reputation that they always start their talk with an apology. So since I'm both, I start my talk with an apology for the following joke. So not so long, I went to a meeting and I checked in a hotel, and the guy at the reception asked me, "What do you do?" And I said, "I'm a cell biologist." And of course I said that very proudly. And then the guy was quiet for a while, and then he said, "What do you sell?" Okay, so what I'm going to sell today is, so I'll give two talks. The first talk is going to be about protein translocation, and then the second talk is going to be about EROD, ER-associated protein degradation. And I invite you to ask questions if you have questions in between. I will make stops at certain points and just ask anyway whether there are any questions. So, but you can ask me in between as well. So let me start out by reminding you that what I'm going to talk about, which is protein translocation. This is the very first step in the biosynthesis of a large class of proteins, which includes proteins along the secretory pathway, for example, in the endoplasmic reticulum, in the Golgi, in the plasma membrane, and it also includes proteins that are exported from the cell that we call secretory proteins. In all these cases, proteins are initially transported across the ER membrane, the endoplasmic reticulum membrane, and they either cross completely to end up in the lumen of the ER, or they are integrated into the membrane, meaning that some segments of the proteins end up in the lumen and others stay behind in the cytosol. So this is the major export pathway in a cell. About 30% of all proteins in every eukaryotic cell are actually exported. And in some cells, it's much more. So once you have done the translocation, then there's a second phase, which is called vesicular transport, which Jim Rothman will be talking about later in this course. So you get a budding of vesicles from the ER that contain the soluble protein inside and the membrane protein in the membrane, and then these vesicles fuse with the Golgi initially, deliver the content into the Golgi and the membrane protein into the membrane. And then by successive vesicular budding and fusion processes, the vesicles eventually arrive at the plasma membrane, and then again the content proteins will be delivered to the outside, and the membrane proteins become membrane proteins. And you also have proteins that, as I said before, stay behind in the Golgi or in the ER, or are diverted to endosomes and lysosomes. So what I'm going to talk about is this very first step, the translocation step. This protein translocation across the ER can be divided into two steps. The first step we call the targeting phase, during which the proteins are directed to the ER membrane by virtue of hydrophobic sequences. And these can be cleavable signal sequences, as in the case of most secretory proteins, or non-cleaved transmembrane segments, as in the case of most membrane proteins. And this targeting phase often involves the function of the signal recognition particle, SRP, and its membrane receptor. And then subsequent to the targeting phase, there's a second phase, which we call the actual translocation phase. And during this process, proteins are moved through a protein conducting channel, either end up on the other side of the membrane, as in the case of a secretory protein, or in the case of a membrane protein, when a hydrophobic segment, shown here in yellow, arrives inside the channel, it moves sideways out into the lipid phase and generates extracellular and intracellular segments of the membrane protein. So this channel has a very unusual property that is not shared by channels that transport small molecules, which only open perpendicular to the plane of the membrane. This channel opens in both directions, across the membrane and also within the membrane. So it's a very unusual kind of channel. The there's a very similar translocation process in bacteria and archaea. So this is a very conserved system that is probably as soon as the cell was generated, we had a translocation system as well. So in bacteria, there's no internal membranes. And so in this case, translocation occurs through the plasma membrane. So in the case of a secretory protein, the protein ends up in the space between the inner and outer membrane in the case of gram-negative bacteria, or in the case of gram-positive bacteria, which only have one membrane, the protein ends up on the outside in the medium. So as you probably know, signal sequences have a very simple structure. They have a usually positively charged N-terminal domain or N domain, then a hydrophobic segment, the H domain, and another hydrophilic segment called the C domain, and then there's a cleavage site. The important part is this H region, which is essential for its function, and it comprises about 7 to 14 hydrophobic amino acids. And the important point is that this sequence, the amino acid sequence of the H region, doesn't really matter. All that matters is that you have a stretch of hydrophobic amino acids. The signal sequences are distinguished from transmembrane segments in that the transmembrane segments are usually longer, about 20 hydrophobic amino acids. But again, the sequence is irrelevant. It's only hydrophobicity that matters. And this has been actually quite puzzling to people for a long time. How can signal sequences be recognized despite the fact that they're so diverse in sequence? Is it protein-protein interaction? Is it protein-lipid interaction? Is it both? The channel is formed from a very conserved, evolutionarily conserved membrane protein complex that consists of 3 subunits. It's called the Sec61 complex in eukaryotes and the Sec-Y complex in bacteria. Here are the names of the subunits. So in eukaryotes, it's Sec61 alpha, beta, and gamma. And in prokaryotes, I'd like you to remember the names because I will be mostly talking about prokaryotes. Sec-Y is the alpha subunit, Sec-G the beta, and Sec-E the gamma subunit. So, and I should also say that only the alpha and gamma subunit are actually essential. The beta subunit is dispensable. Now here's a very important point. The channel itself is just a passive pore. It doesn't transport proteins on its own. It needs partners that provide a driving force for translocation. And we have different modes of translocation. There's this co-translational mode of translocation, which means that the polypeptide chain is transported across the membrane while being synthesized. And there's a post-translational mode of translocation, and also in all cells, in which the protein is first completed in the cytosol and then translocated. And post-translational translocation is done differently in eukaryotes and prokaryotes, whereas co-translational translocation is universal. Now you might wonder what is the distinction between these two pathways, and it turns out that the more hydrophobic signal sequences go into the co-translational pathway because they interact efficiently with the SOP while they're being made on the ribosome. So let me say a few words about the 3 modes of translocation. So first, the co-translational mode of translocation. It's also responsible, this is the most general mode, and it's also responsible for the synthesis of most membrane proteins. So the mode is very simple. The ribosome sits on top of the channel such that the tunnel inside the ribosome through which the polypeptide chain is moving is aligned with a pore in the membrane. And so essentially, the polypeptide chain has only one way to go. And the energy for translocation comes from the energy of translation, which is GTP hydrolysis. So you don't need any additional energy. The second mode is post-translational translocation, which was worked out in yeast by a former postdoc in the lab, but it's probably applicable to higher eukaryotes as well. So in this case, the channel, again here in red, associates with another membrane protein complex, which in yeast is a tetramer. It's called the Sec62-63 complex. And you also need a luminal protein called BIP. BIP is a member of the HSP70 family of ATPases. It's a chaperone. And it acts as a molecular ratcheting molecule to move the polypeptide chain across the membrane. This slide shows it in more detail. So BIP starts out in the ATP bound state. And as all HSP70s, in the ATP bound state, it has an open polypeptide binding pocket. Then it interacts with one domain in Sec63, which is a J domain, which induces rapid ATP hydrolysis. And so the peptide binding pocket closes as the molecule goes into the ADP bound state. And it closes around the incoming polypeptide chain. So now the BIP is bound to the polypeptide chain, and it's too big to fit through the pore of the channel, so it prevents the chain from going back into the cytosol, but it does not prevent it from going forward into the lumen. And if it has when it has moved a sufficient distance into the lumen, then the next BIP molecule will bind, and so on, until the polypeptide chain is all the way through. And eventually you get nucleotide exchange, ADP goes out, ATP comes in, the peptide binding pocket opens, and you fall off. And you get a free polypeptide chain ending up on the luminal side. The third mode of translocation is post-translational translocation in bacteria. In this case, there's no ADP on this side, so the mechanism I just explained wouldn't work. And you have instead an ADPase on the cytoplasmic side. It's called Sec-A, and it uses the energy of ATP hydrolysis to push the polypeptide chain through the channel. So in summary, we have three different modes of translocation. The channel, here in red again, is always the same, more or less. But the partners are different, and the way the driving force is provided is completely different in the three different modes. And so it's an interesting modular system. So I'll be talking for the rest of the talk about the bacterial system, which is, for several reasons, is easier to study. Yes, sure.
The BIP binding of the polypeptide, is it done at any particular residue or
No.
any residue?
No, actually BIP is very non-specific once it's activated by this J protein, this J domain on Sec63. So it binds to any incoming polypeptide chain. It's different from the binding that normally, if you just interact BIP with peptides, you find that it prefers hydrophobic sequences. But once it's activated with J, then it binds non-specifically, even though they don't fit perfectly into the binding pocket. Any other question? Yep.
So after the translocation again by BIP, what will make the BIP the main release the peptide? Otherwise it will stay
Nucleotide exchange. So it happens spontaneously. So the ADP goes out, ADP comes in, and then the peptide binding pocket opens, and you fall off.
So how is fat integrated in the membrane in the first place, or especially how the orientation is?
So, okay, it's a good question. So initially, the polypeptide chain is inserted as a loop, with the single sequence being one part of the hairpin, and the mature region following the single sequence being the other part of the hairpin. I'll come back to this point later. Okay, let's go on. So let me tell you first a little bit more about the channel itself. So about 14 years ago, or 12 years ago, we were able to get the first crystal structure of these Sec-Y channel, so from an Archaeobacterium, Methanococcus yanashi. And these are the three postdocs that did the work, and this was a collaboration with Steve Harrison. So what you see in the upper panel is a top view. So you're sitting in the cytosol and you're looking down onto the membrane. And I think you can appreciate that the alpha subunit, the largest subunit, consists of two halves, shown here in blue and red. Transmembrane segments 1 through 5 in blue and 6 through 10 in red. And they're linked at the extracellular side by a loop between TM5 and 6. So the gamma subunit wraps around the two halves and keeps the two halves together. The beta subunit sits on the side and makes relatively few contacts with the alpha subunit, and so it may explain why it is dispensable. So this side we call the front side, and the molecule can open up on this front side like my fingertips do. So this we call the lateral gate of the channel, and you can immediately appreciate that this is the side where the transmembrane segments would exit laterally into the lipid. The lower panel shows a side view, And you can see that the channel has an hourglass shape with an aqueous funnel on the cytoplasmic side and also on the extracellular side. The cytoplasmic funnel is empty, and the extracellular funnel is filled with a little helix that we call the plaque. And in the center, there's a constriction formed by 6 hydrophobic amino acids that project their side chains radially inwards. So we call this the pore ring. In E. coli, all 6 pore residues are isoleucines. And in other organisms, there are also aliphatic residues that have usually branched side chains. So the crystal structure of this closed inactive channel, this was crystallized in the absence of a channel partner, in the absence of a translocation substrate, led us to models of how translocation really works. And I have to say, most of these predictions turned out to be correct. There was one exception where we were wrong, or partially wrong, I should say. So the first thing that we predicted is that this plaque, this is again the top view, and the plaque is here colored in green. We postulated that the plaque would have to move out of the way to open the channel, shown schematically here. So the plaque would move out of the way, and this would open the channel so that you could move a polypeptide chain from where you're sitting into the board. So this would, the polypeptide chain would move through the aqueous funnel in the cytoplasmic side, through the pore ring, and then on to the extracellular funnel, and then further on into the extracellular space. So there's a few points that I would like to make here. First, the polypeptide chain would move in an aqueous environment. And of course, this minimizes the energy that you need to move a hydrophilic macromolecule through a hydrophobic membrane. And the second point is that the hourglass shape of the channel and the hydrophobic nature of the pore residues would minimize contact of the translocating polypeptide with the channel. And again, this would reduce the energy that you need to move the polypeptide chain across. I'll come back to that point. Yeah, that's a good point. And then finally, we postulated that once a hydrophobic segment would arrive inside the membrane, so it would move from into the board, it would move out sideways through this lateral gate, shown here in yellow and blue, into the lipid phase and become a transmembrane segment. If you look at the lateral gate from the side, you can see that it consists of 4 little segments or 4 different transmembrane segments, so it is actually a weak seam. And in fact, later X-ray structures from different species show this lateral gate partially open. So it is a very weak interaction across the gate. So the next big step in this field was when we were able to get a structure of the sec-A, sec-Y complex. This was a different species. I should have said that archaea don't have sec-A. We still don't actually know how post-translational translocation is done in archaea. But in bacteria, this is a bacterium, a thermophilic bacterium, and they do have sec-A. And this was the work of 2 postdocs, Jochen Zimmer and Yunsen Nam. So this shows a side view of the structure. This is sec-A. As I mentioned, is an ADPase, is a flat molecule that kind of lies with a flat surface on the membrane, parallel to the membrane. The gray stuff is sec-Y. This is sec-G, the beta subunit, and in purple is sec-E. If you look from the top, you can appreciate that sec-A has different domains. There are 2 nucleotide binding domains, MBD1 and 2, and the nucleotide is bound in between. And so during the ADP hydrolysis cycle, the 2 MBD domains move relative to one another. By the way, this is a Rec-A-like fold in each case, so you may know that there are other ADPases, usually hexamers, that use the Rec-A-like fold. So then following, this yellow domain makes most of the contact with the sec-Y molecule. This long helix we think is like a lever arm that amplifies the motions here towards the region towards this, at the C-terminus. But the most important feature that we noticed when we looked at the X-ray structure is this, what we call the 2 helix finger. there's a helix, a loop, and a helix going back out. And this 2 helix finger pokes into the cytoplasmic funnel into the Sig-Y channel. And so this gave us some ideas of how the Seg-A ADPase may actually move polypeptide chains through the channel. So how does Seg-A move a polypeptide chain through the channel? I think that suggested a mechanism. And so the mechanism that we suggested is that the 2 helix finger would move up an ATP binding towards the polypeptide chain, bind to the polypeptide chain, and then move towards the channel, and thereby drag the polypeptide chain with it and push it into the Sig-Y channel. And this cycle would then be repeated. Turns out, and I'm not going to show you any data on this, that it's a little more complicated. And we could have thought about it right away, but it took us quite a while to actually realize that this is not as simple. And the reason is that this loop that interacts with the polypeptide chain, which has a tyrosine at the fingertip, which is also typical for many of these ADPases, it doesn't interact with every amino acid equally well. For example, if you have a glycine at this point here, it actually doesn't interact at all. So it will move towards the polypeptide chain and move down, but nothing will happen because it doesn't interact with the polypeptide chain. And so there are only some interacting hydrophobic amino acids, shown here in red, and there are other amino acids, for example, like glycine residues, in which case there would nothing happen. And so what is important is that there's also sliding of the polypeptide chain, passive Brownian motion within the channel. So in the case that there is a glycine residue, the polypeptide chain can slide back and forth. And in the ADP-bound state, where the finger is away from the chain, the sliding can happen with any amino acid that is present in the channel at this point. So we call it the push and slide mechanism. In other words, the finger gives occasionally a push to the polypeptide chain in the right direction, but most of the time the polypeptide chain would slide back and forth. So imagine you have a glycine stretch of 20 amino acids. So the finger cannot do anything, but it can slide, for example, backwards. In this case, you would just start over again. Or it can slide forward, and then you have amino acids behind the glycine that would be able to be pushed into the channel. So this is what we think is a mechanism by which the ADPase can translocate any sequence. And it's important, obviously, that you can translocate any polypeptide sequence, regardless of whether you have interacting or non-interacting amino acids. Any questions? Yeah, so it's a good question. We don't actually know. It turned out to be very difficult to identify which amino acids it interacts with and which it doesn't interact with. So we still don't know. We know glycines is bad. What we haven't really done is very systematic analysis of which amino acids are interacting. We assume that the bulkier residues are the ones that are the best interacting ones. Vivek? Sure. We did a mathematical model, and we can calculate the rates depending on the number of interacting amino acids. And of course, you have a lower rate if you have less frequent interacting amino acids. But it's actually not, translocation is a very slow process. It takes about a minute or so to translocate them in, let's say, 200 amino acids. It's incredibly slow. And if you think about the co-translational mode, the rate of translocation is determined by rate of translation. And you make about 20 amino acids per second in a normal eukaryote. It depends a little bit of eukaryotes are a little slower than prokaryotes, but it's still very slow.
So then in that case, you need these 2 helix finger or peptides that are distinguished from glycine.
Well, you need it to get directionality. If you don't have the finger, you don't get directionality.
So there must be some evolutionary pressure, right?
Right. If we knew exactly which amino acids were interacting and not, then maybe we could do that analysis, but so far we can't. I mean, you don't find 20 glycines in a protein that is translocated, so that far can go, but I cannot really tell you.
Have people done mass experiments, for example, in the medium, the protein type, secretly tend to see you have more glycines, you have slower breaking.
Right. So, I mean, we checked whether any secretory protein would have a stretch of long glycines, and it doesn't happen. So we can engineer. We did that, and then we find that translocation is much slower. You know, but you know, it's not dramatically slower.
Why the polyglycines also prevent insertion of the substrate into the proteome? Is there any kind of stimulation mechanism?
Yes, in some ways, yes, because remember in the proteasome you have this ring of ADPases, and these ADPases each have a Rec A fold, and each of them have an aromatic residue that interacts with the polypeptide chain. So in this case, you have 6 loops, like the 2 helix finger, that interact with the polypeptide chain, but it's very similar. And all these Rec A-like ADPases that transport proteins have this aromatic residue at the tip, and they all have the same problem. I believe that in the Sec A doesn't really have a lot of power to unfold polypeptide chains. So these hexameric ADPases generate much more power. So that's the major difference.
Do you have
So
structures of Sec A both in the HT bound state and the interstate?
no, unfortunately we don't. The reason is that the interaction between the Sec A and the channel is much weaker in the ADP state. Yeah. Okay? All right. So very recently, at the beginning of this year, we were able to get yet another structure, which has been a dream for us for a while, namely the crystal structure of the active channel. That is a channel which has a translocating polypeptide chain inside the channel. And this is work by 2 postdocs in the lab. Well, actually, 1 postdoc, Long Lee, and the other one is a former graduate student, Union Park, who then became a postdoc with Rob McKinnon's lab. Okay, so let me tell you a little bit about this structure. So the goal that we had was to generate a structure that looks like this here. So it would consist of the Sec-Y channel. This is the 3 subunits that I mentioned. It would consist of the Sec-A ADPase and a substrate. And the substrate would have a single sequence that is of lower hydrophobicity, so that it would go into the post-translational pathway. And then at the C-terminus, we would put a folded domain. This is a fast folded GFP, so that it would form a blob at the C-terminus that could not be moved through Sec-A and the Sec-Y channel. So this would generate this loop insertion. Somebody asked me earlier how the polypeptide chain inserts into the channel to initiate translocation. So the single sequence would sit on one side of the hairpin, and then this would be the region that would be normally the cleavage site, but we mutated away the cleavage site so it would not be cleaved. And then you would have this blob at the C-terminus that would generate a translocation intermediate. Okay? So what we tried initially is to express in E. coli the Sec-Y channel, the Sec-A protein, and the substrate, and to assemble this intermediate and then purify it and try to crystallize it. And we can purify milligrams amounts of this complex, but unfortunately it never crystallized. So then Union Park, the student, had this great idea to open the 2 helix finger at the tip and insert a linker, the single sequence, and the mature region of the polypeptide chain, and then go back into the second helix of the 2 helix finger. So this looks like this here. Here's Sec-A, here's a 2 helix finger, here's the substrate inserted into the Sec-A molecule. So now you reduced it to 2 components rather than 3 components, and obviously this simplifies the system. Now we put a cysteine in the polypeptide chain after the single sequence, and another cysteine into the SIG-Y channel, so that when the polypeptide chain inserts into the channel, these two cysteines would be on the periplasmic side and could form a disulfide bridge. And this would indicate that the polypeptide chain is actually inserted into the channel. And this surprisingly actually works. So here's a cross-link. If we introduce here into the hydrophobic single sequence a few hydrophilic residues, we lose the cross-linking between the SIG-Y and this fusion protein. So we also switched species here. So we're using SIG-A from Bacillus, and we're using the SIG-Y complex from a Geobacillus species. The reason is that this is a compromise between thermophilic. As you probably know, for crystallography, you always try to use a thermophilic organism because it's more stable. But the problem is we need to express it. It needs to be active in E. coli. And so you don't want to if you go too high with the temperature, it will not be active at 37 degrees, which is the gross temperature of E. coli. So we use the compromise. This organism grows at about 50 degrees. So what we do is we co-express in E. coli the SIG-Y-E complex. We left out the SIG-G because it's not needed. And then also express this fusion protein between SIG-A and the substrate. We also have a GFP at the C-terminus. Then we do the cell lysis, isolate the membrane, solubilize, pull on the his tag, which is on this SIG-Y complex, and then cleave off the GFP, do a monocuse chromatography, size exclusion chromatography, get a pure protein, and crystallized. And so I actually have to also say that it wasn't quite as simple as I just explained. We also had to raise a nanobody. This is a single chain antibody made by camels and other, in this case it was an alpaca. So you have the nanobody bound to the periplasmic side, so that gives you more crystal contact and allows crystallization. So here's the structure. So this is SIG-A again. The brown thing here is SIG-Y, and the purple is SIG-E, and the green is the most important thing, which is the translocating polypeptide chain. The helix here is the single sequence, and the part that you see in the back, in the middle of the channel, that's the translocating polypeptide chain in the pore proper. So now we have three different structures. We have the idle structure from Methanococcus, we have the SIG-A bound structure, and we have the active channel now with the translocating polypeptide chain. Now they're different species, but we think that we can still compare them because actually the channel looks quite the same. So let me first tell you a little bit about the plug. So in the closed state of the channel, the plug is in the center of the molecule and basically blocks everything. So this is a closed state of the channel. Nothing goes through the central pore. In the SIG-A bound state, the plug actually moves towards the front. The lateral gate opens up a little bit. I'll show you this in a minute. And the plug sits in the front and prevents, presumably in a native membrane, the influx of lipids into the channel. And in the active channel, the plug is moved towards the back. Now this looks like an open lateral gate, but in fact the single sequence is sitting here, and I left it out here. So the lateral gate in the idle channel, the closed state of the channel, is completely closed. There's these three transmembrane segments form the lateral gate, TM2, TM3, and TM7. In the SIG-A bound state, the lateral gate opens up. I think if I toggle back and forth, you can appreciate that. So it opens up. The distance here becomes larger. And if you look at the active state of the channel, there's really not that much difference, except that these two come closer to one another. And as you will see in a second, this actually forms like a groove on the outside of the channel. So the most important thing that we wanted to understand for this structure is how is the single sequence bound? How is a single sequence recognized? And to our big surprise, that's even more surprise. What's happened? Yeah, it's just a bad connection here. Okay, good, fixed. All right, so to our big surprise, it turns out that the single sequence sits outside the lateral gate. So here's the N domain, the positively charged, and in an intact membrane, it would interact with the head groups, the negatively charged head groups of the phospholipid bilayer. The hydrophobic H region sits outside the lateral gate, and in a native membrane, it would face mostly lipids. And then the C region is intercalated into the lateral gate. It sits between two transmembrane segments. The cleavage site is shown here with the blue ball here. Turns out to be right inside the channel, and it would not be accessible by the single peptidase that cleaves off the peptide. So we have to assume that at some point there will be a conformational change in the single sequence that would move this cleavage site towards the extracellular space so that single peptidase can cleave it off. So it turns out that there also interaction between the hydrophobic region and the outside of the transmembrane segment 2. So these are hydrophobic interactions, van der Waals contacts, and we believe that this means that there is some recognition, protein-protein interaction, that may explain why some amino acids occur more frequently in the H region than others. But the major conclusion is here that the single sequence is recognized mostly by lipid interaction. And but there seems to be some contribution from interaction with the channel itself. And so we believe that this explains why single sequences are diverse and why only hydrophobicity matters, and why nevertheless certain amino acids occur more frequently than others. So for example, leucine is preferred over isoleucine and valine, despite the fact that they all three have about the same hydrophobicity. So you might ask me, how do we know that our structure is not an artifact of the heavy engineering? After all, we've fused the substrate into Sec-A. We had a disulfur bridge here. We used nanobodies. What I didn't tell you is that we had to use heavy metal ion cluster to actually did the phasing. So this is not a molecular replacement structure. This is actually real phasing. So luckily for us, there was also a structure published at the same time with cryo-EM of a mammalian ribosome nascent chain Sec61 channel complex. This was the work by Rebecca Voorhees and Manu Hector. And they also generated the complex in a completely different way. Now, again, I think it's the movies that it doesn't like movies. Yeah, okay. But it turns out that the structures are actually almost superimposable. So their structure was of lower resolution, so they couldn't actually determine the registry of the helix. But ours is here in green and theirs in yellow, and you can see that the two helices basically overlap completely. So in both cases, the single sequence sits outside the lateral gate. For some reason, these guys actually didn't point it out. But I think they kind of missed the most important point, but the single sequence sits at the same side. And moreover, there's an earlier structure at much lower resolution by Roland Beckman, where they looked at the nascent membrane protein, and they found that, again, the transmembrane segment of this nascent membrane protein was also at the same side. And so what I find the most gratifying conclusion is here, single sequences and transmembrane segments are recognized in the same way in all organisms and translocation modes. Namely, hydrophobic segments exit the lateral gate, dock into a groove at the outside where they encounter the lipid phase, and presumably it's the lipid interaction that is most important and explains why single sequences and transmembrane segments are so diverse. Any questions? Not in the, okay. So in the eukaryotic system, it doesn't matter. In the bacterial system, you need negatively charged lipids. We know that this is in part because seg-A binding to the membrane requires negatively charged lipids, but it's not the only thing. We can artificially tether seg-A on the membrane, we still need negatively charged lipids. And we don't really understand why that is.
You showed us before that the proteolytic side is a little bit
Buried.
problematic, so how does the signal peptide provide this accessibility?
So there is some data from with synthetic peptides that suggests that there is a conformational change that may expand the helix, so that maybe converting from an alpha helix to maybe a 310 helix or something that would move the cleavage side more towards the extracellular side. But I mean, what I would really like to have is a structure where this, maybe with a longer polypeptide chain. It's not so easy, but that would be the ideal thing. They're important for the initiation of the translocation process. So you need this hydrophobic segment to get this loop insertion to start with. Then for the further translocation of the polypeptide chain, you can get sliding back and forth. So eventually the polypeptide chain will go through anyway if you don't have anything on the other side. So no blob on the other side. But in order to make it more efficient, you need to have some driving force like the ribosome or seg-A or something on the other side like BIP. Okay?
Why does it insert the right way around?
So the major thing we believe is this positive charges at the N-terminus. So when they come out, they interact with the negatively charged lipids, and this it retains the N-terminus on the cytoplasmic side. Now, you might ask me, there are membrane proteins that have the opposite orientation, where the N-terminus goes across. In that case, there are no positive charges at the N-terminus. And so it's believed that the N-terminus flips across the membrane right away. And that will terminate translocation right away. And so then it moves sideways out. So the final point I would like to make on the structure is how does a translocating polypeptide chain look like in the actual channel? And so this is the polypeptide chain inside the actual pore. So here's a single sequence sitting on the outside of the lateral gate, and this is the mature region of the polypeptide chain. So during translocation, this would be the part that moves through the membrane, whereas this one stays kind of constant, stationary until it's cleaved off. Now you see that there's an angle of about 45 degrees between the single sequence and the mature region. And this guarantees that there's no interference of the single sequence with this mature region of the polypeptide chain. So there's no interaction between the two parts of the hairpin. And you can also see that the polypeptide chain is in an extended conformation when it is moving through the channel. So no secondary structure here. And finally, what you don't see here, but I can tell you, is that the only contact between the translocating polypeptide chain and the channel is with the pore ring. So this is what we predicted, that the polypeptide chain would only interact with the pore ring. There's no interaction in the cytoplasmic funnel or in the extracellular funnel.
And again, can you see the time resolution to understand why protons don't leak?
I'll come back to that in a second, and actually indeed in a second here. So here it is. So if we look at the idle channel, the closed state of the channel, you can see that the plaque and the pore ring completely seal the channel. This is shown here in the space filling thing, network, meshwork. So no small molecules even could go through here. this is a pretty good seal. In the open channel, in the active channel, the plug is moved out of the way towards the back. This is the yellow thing. And the translocating polypeptide chain now sits in the channel. And you can see that out of the 6 pore ring residues that were here in the closed state of the channel, 2 are displaced. And the 4 remaining ones sit around the translocating polypeptide chain like a gasket. So they basically seal around the translocating polypeptide chain. And this prevents the free flow of ions while you're transporting a translocating a polypeptide chain. If you think about it, I mean, Roger was pointing it out probably, it's not trivial to translocate a macromolecule through the membrane and yet prevent small molecules from going through at the same time. And so that nature has found a really elegant way to deal with that. So here's a scheme to explain the mechanism again. So in the closed state of the channel, you have the plug and the pore ring sealing the channel. And I should say this is not just based on this X-ray structure. Unyong had actually done a lot of experiments in vivo, experimental work, to look at the influx of small molecules into the cell. So in the closed state of the channel, the pore ring and the plug seal the channel so nothing can go through. In the open state of the channel, the plug is out of the way, but now the translocating polypeptide chain and the surrounding pore ring residues seal the channel. When the polypeptide chain is either all the way across to the other side, or if a transmembrane protein is made, you move sideways out into the lipid, the plug can return to the center of the channel and reseal the channel. So this means that at all times, there is a barrier for the small molecules to go across. Now, I would admit that this is not a perfect seal. So there are protons, I'm sure, can go through quite frequently. But the cell has very powerful pumps that will correct this flow of protons and therefore maintain viability. We actually, so Unyang mutated some of the pore ring residues. If you mutate one of these isoleucines to glycine, you're still okay. The cells grow slowly, but they're still viable. If you mutate 2 to glycine, you're dead. So and then we can show that depending on the number of glycines that you have in this that you mutated to, you can move with 1 glycine. You can move still some small molecules, but not the larger ones. With 2 glycines, you can move more larger things across, and so on. Okay, and the other conclusion from this structure is, let me go back again, is that there is an opening towards the lateral gate, right? So the pore ring residues surround the translocating polypeptide chain, but there is a gate here that allows for example, hydrophobic lipid chain, hydrocarbon chains to contact the polypeptide chain. And what we therefore postulating is that the hydrophobicity of the segment inside the channel will be continuously probed. And if the hydrophobicity is high enough, then the polypeptide chain can move sideways out into the lipid phase and become a transmembrane segment. So this is a completely passive partitioning process that only depends on the segment, the 20 amino acids that are at any given time inside the channel. And so this is my final slide where we basically, on the basis of these structures, can propose a mechanism. So we're starting with the idle channel. Then the Sec-A protein binds. This opens up the lateral gate to some extent, not entirely, but it's a open, partially open gate. Now the translocating polypeptide chain inserts as a loop with the positive charges retaining the N-terminus on the cytoplasmic site. The polypeptide chain inserts as a loop, and the signal sequence moves out of the lateral gate and docks on the outside into this groove with the hydrophobic amino acids facing the lipid environment. And then at some point, there's a conformational change that leads to cleavage, and then the polypeptide chain is moved through the central pore in an extended conformation towards the outside. And with that, I'm going to finish my first lecture and just want to acknowledge the people who did the work. The most recent work, this push and slide mechanism was done by a former student in the lab, Benedict Bauer, and the more recent structure of the active translocon was done by Long Li and Eun-Young Park. And we got help from Hide Plus lab. They were the ones who raised the nanobodies that we use for the crystallization. Thank you so much. So we have plenty of time for questions. Yeah.
I still didn't understand how the directionality of the SAC complex is controlled. So there's a polarity.
I'm sorry?
You know, why SAC complex is inserted this way rather the other way around. I still can't understand.
Okay. Well, I have some slides here for. It's basically, if I understand correctly, you're asking me about membrane protein insertion and how you get the orientation of the membrane proteins.
Especially the SAC complex you are talking about, it is translocation.
I'm not quite sure I understand the question.
This channel, so why is this way but not the other way? Otherwise, you will pump the protein in a wrong direction.
He's asking why the channel itself is
Why the channel itself?
Why the channel itself?
Yes.
Well, I mean, the channel is a membrane protein, so it actually is integrated by the same translocation machinery.
It's the first molecule of the
Yes, yes. But this is like the hen and egg problem that we always face, right? Cells are only generated from cells. Remember this old sentence by Rudolf Virchow, omnicellular, acellular, meaning cells are only generated from cells. And the same is true here for membranes. Membranes are only generated from membranes. They're not generated de novo. This sec-Y complex requires a sec-Y complex for its integration, for its new synthesis. But since I misunderstood here, but this is basically how we imagine the different types of membrane proteins to be synthesized. And in some, I think this is pretty established, but some of the issues about membrane protein integration are still a little bit shaky. So here we're looking at what's called a type 1 membrane protein. Those are membrane proteins that have a cleavable signal sequence and a downstream transmembrane segment. So here's a ribosome synthesizing the polypeptide chain. When the signal sequence has emerged from the ribosome, you get this loop insertion that I just talked about. At some point, you get signal sequence cleavage. And then when the polypeptide chain, if there wasn't any further transmembrane or hydrophobic sequence, the polypeptide chain would end up on the other side. But if there is another hydrophobic segment shown here in yellow, then the polypeptide chain, when it arrives inside the channel, would move out sidewise. The subsequent segment would emerge into the cytosol between the ribosome and the channel. And then finally, they have the C-terminus here and the N-terminus here. So it's a single spanning membrane protein that you with this topology.
Are you going to show a multiple spanning?
Yes.
You explained that the signal sequence sort of enters this pocket and is stuck there until the whole protein, the whole peptide goes by. But then once it's cleaved, how does it get released from this pocket?
That's a good question. So there's a single peptide peptidase that cleaves the single peptide once it's cleaved off from the polypeptide chain. And this peptidase is a presenilin-like peptidase. It cleaves inside the membrane. And then you get two segments of the signal sequence, and both of them are not hydrophobic enough to stay in the membrane, so they're released.
Supposing you engineer signal peptide followed by polyglycine, and then downstream you engineer vector amino acids. Whatever you choose, can you control the speed or the kinetics when you insert, and then you also gain information about the amino acids, enzymes, associative?
Yeah, it's not as easy as you think. Yeah, in principle, one can try that, but we haven't.
You have tried it?
We have not. We put the glycine residue further downstream, not immediately after the signal sequence.
So what happens if you have a protein that has the N-terminus?
I'll show you in the next slide.
So what happens with tail anchor proteins?
Oh, okay. Let me first finish the membrane proteins of the other orientation, and then we'll come back to the tail anchored. All right, so the next class of proteins is called type 2, or some people call it single anchor type 2 protein. So in this case, you have at the N-terminus a hydrophobic segment, but it's longer than a cleavable signal sequence. So these are non-cleaved hydrophobic segments. But you also have, like in the case of a signal sequence, a positively charged N-terminus. So what happens is that the polypeptide chain again inserts as a loop with the positive charges retaining the N-terminus on the cytoplasmic side. But in this case, the hydrophobic segment is hydrophobic enough to leave the channel all the way into the lipid phase, like as shown here. And so then all you do is complete the polypeptide chain so that the C-terminus ends up on the luminal side. And so you get this orientation here with the N-terminus in the cytosol and the C-terminus on the luminal side. That's type 2. Type 3 is also called single anchor type 1. And in this case, the positive charges are on the other side of the hydrophobic segment. And there's no positive charges to retain the N-terminus. And often these hydrophobic segments are longer. And this segment here also needs to be unfolded, or at least loosely folded. And so what happens is that this N-terminus flips across the membrane right when the polypeptide chain is inserted into the membrane, and it moves also sideways out into the lipid phase. And then the polypeptide chain is completed by the ribosome, and so you get this opposite orientation with the C-terminus in the cytosol and the N-terminus on the luminal side. And the only other class I need to explain is multi-spanning membrane proteins. And I just picked one example here where you have 3 transmembrane segments with the N-terminus on the cytosolic side and the C-terminus on the luminal side. So you again start, let's say, with a single anchor type 2. So the positive charges are in the N-terminal side. You get loop insertion. Then if nothing happened, you would get, the transmembrane segment exits the channel into the lipid. And if nothing happened, you would get just a single spanning membrane protein. But if there is another membrane segment, then it arrives inside the channel and you move out sideways. And now you would, if nothing happens, you would get the C-terminus on this side. But if there is another hydrophobic segment, you get loop insertion in the same way as you got it at the beginning. So the hydrophobic segment would then reinitiate translocation. This segment would move out eventually, and you get the C-terminus on this side. Okay? Any questions about this? Yeah?
On all of these examples, single sequence stay uncleaved?
In all these, except for the type 1 that I mentioned at the beginning, there's a single sequence cleavage. Yes. That's actually not very common in membrane proteins. So most membrane proteins don't have a cleavable single sequence.
How long does the topic start to be?
20, approximately 20.
So each segment is kind of probed out independently, or is it really a combination of everything that is already in the membrane
Right.
that can attract?
That's a good question. So there are examples where 2 neighboring transmembrane segments interact with one another and move out as a pair. Yes, that's it's known that some cases.
Pretty probably very question, but you said only hydrophobicity matters, but does it at least have to be a glycol?
So, yeah, so with multi-spanning membrane proteins, as you probably know, many of them form channels with an aqueous interior. So many of these channels have hydrophilic residues that are inside the membrane eventually. So for example, voltage-gated channel have 4 arginines in one of the transmembrane segments that respond to the membrane potential. So the question is, how do you insert a transmembrane segment that has so many positive charges? It's actually very unstable in the lipid bilayer. 1 is tolerable, 4, no way. And so it's not really clear how this works, but one idea is that on the outside of the channel, on the outside of the lateral gate, there is a membrane chaperone that accepts the transmembrane segments as they move out and transiently keeps them in the membrane until all the other transmembrane segments have been synthesized and can form a structure that is hydrophobic on the outside and hydrophilic on the inside. So these membrane chaperones are not really, it's not very established what these membrane chaperones are, but in the case of E. coli, it's called YIT-C, likely, and in the case of mammals, it's called a protein, we call TRAM. Although I have to say this is still very speculative at this point.
Signal peptide have to be alpha-helic.
Yes, so I mean well, I mean this is the only structure, the only structures I should say.
Is that only hydrophilic matter? So that's why
Well, we think, so you can, so Chris Kaiser and David Botstein did a very famous experiment where they took random sequences and put them in front of an invertase without a single sequence and asked what's the percentage of sequences that would work as single sequences, and it's amazing. It's like 30% of all sequences work to some extent. And if they look at the sequences, it really only hydrophobicity. So I think when you're hydrophobic, it works to some extent, but there's obviously optimization, and the actual single sequences are more hydrophobic than these random sequences. Should probably start with the next talk, huh? Okay? Ready? Okay. So now I'm going to talk about EROD, which stands for ER-associated protein degradation. So I'll come back to the acknowledgement period later. So far, we've been talking about this process, the translocation of a protein across the ER membrane into the lumen. And as you probably know, proteins undergo quality control once they're translocated, such that only properly folded proteins are moved on along the secretory pathway. If a protein does not reach its native folded state, it will eventually be transported back into the cytosol. So it will move across the ER membrane in the reverse direction, so this is why we call it retrotranslocation, will then be polyubiquitinated on the cytoplasmic side and eventually be degraded by the proteasome. And this is the process I'm going to talk about. It's called ER-associated protein degradation, or EROD. EROD is conserved in all eukaryotes, and it's not only involved in the degradation of misfolded ER proteins, it's also involved in the regulated degradations. So for example, there's this HMG chorea reductase, which is degraded when you have sterols in the cell because it's no longer needed. The process is also hijacked by certain toxins, for example, cholera toxin. Cholera toxin binds to the surface of a eukaryotic cell, then moves backwards along the secretory pathway until it arrives in the ER, and then a portion of the toxin moves across the ER membrane into the cytosol, and then it does its bad thing inside the cell, so you get chloride and water secretion and diarrhea. And then finally, it's also hijacked by certain viral proteins. For example, the human cytomegalovirus downregulates MHC class I molecules so that the viral peptides cannot be presented on the cell surface, and the virus can propagate in the infected cells without the cells being destroyed. So work in yeast by a former postdoc in the lab, Pedro Carvalho, who is actually in Vivek's Institute, Barcelona, just moved to Oxford, has shown that there are 3 different ERAD pathways. Actually, he has discovered a fourth pathway, which I'm not showing here in this slide. So, and the three pathways differ on where the misfolded domain is localized. If the misfolded domain is in the membrane protein on the cytoplasmic side, there's a ubiquitin ligase involved that is called DOE10. And we call this the ERAD-C pathway, C standing for cytosol. If the misfolded domain is on the luminous side, either in a soluble protein or in an integral membrane protein, these proteins will use the ERAD-L pathway, and the ubiquitin ligase involved is called HERD1. And the same ligase is also involved in ERAD-M, where the misfolded domain is inside the membrane. On the cytoplasmic side, all pathways converge at the level of an ADPase complex. The ADPase is called CDC48 in yeast and P97 in mammals. And this ADPase pulls the misfolded protein, ubiquitinated protein, into the cytosol, and then it's moved on to the proteasome. So, from systematic pull-down experiments and genetics, we know most, perhaps even all, components involved in ERAD, particularly in Saccharomyces. As I said, from these two types of experiments, but the mechanism remains unknown. And in particular, what is the translocation channel? We believe that, particularly for ERAD-L, you need a channel to move a misfolded protein across the membrane. It's not happening spontaneously. And so we would like to know what the channel is, and I will tell you, we think we have finally identified the channel after we misidentified it twice. Well, not only us, but other people as well. So the goal that I've set for the lab for quite some time now is to reconstitute ERAD with purified proteins so that we can gain mechanistic insight, because all experiments so far have been done really with a rather crude in vivo system, and it's impossible to gain mechanistic insight. So this slide shows how ERAD-L work, or what the components are. So, as I said, we have identified, we meaning the field, almost all components, probably all components that are involved in ERAD-L. So we're starting out in the R lumen with a misfolded protein, and then there is this complex of 5 different proteins in the R membrane. One of these proteins, YOS9, is a soluble protein, a luminal protein, but the rest of these proteins are all integral membrane proteins. And the most important protein of this assembly here, we call it the HERD1 complex, is in fact the HERD1 protein. This is the ubiquitin ligase. It has on the cytoplasmic side a ring finger. This is a large class of ubiquitin ligases. And it also has multiple transmembrane segments. We now know from structural work that we've done recently, we have a EM structure, which I'm not going to show you in this talk, of a complex of HERD1 and HER3. And so we've now found out that this has actually 8 transmembrane segments, and everybody else, including us, initially thought it only has 6. So this complex somehow allows the misfolded protein to move across the membrane. Once it's on the cytoplasmic side, at least a loop on the cytoplasmic side, there will be polyubiquitination catalyzed by the ubiquitin ligase HERD1. And then there's this ADPase complex, CDC48, that will bind the ubiquitin chain and then pull the polypeptide chain out into the cytosol. So we call that membrane extraction. Now, as you can imagine, if you have so many membrane proteins, it's not so easy to reconstitute the whole thing. So fortunately for us, Pedro actually found out that we can simplify the system. We call this basic ERAD-L. I should say we are mostly interested in ERAD-L because we think it's the most complex pathway, and it's also the most challenging thing to move a polypeptide chain across the membrane. If you already have a domain on the cytoplasmic side, maybe it's easier. So what Pedro found is that when you overexpress HERD1, you don't need the other components anymore. whereas all the cytoplasmic components are still required. This makes the cells grow a little slower. The substrate selection is less specific, so you're starting to degrade things that you shouldn't degrade. And it also turns out that HERD1 itself gets polyubiquitinated and degraded. So, but this aside, it still is ERAD. And that's why we call it the basic ERAD process. And this is our first goal. So we want to reconstitute basic ERAD-L. So this now simplifies the system quite dramatically. We only have one membrane protein now to deal with. And we can distinguish different phases. So the first one would be interaction of the substrate with the HERD1 ubiquitin ligase. The next one would be the ubiquitination reaction. And the third one would be the extraction by CDC48. So relatively simple system. But if you think about it, you need still quite a number of components. So we need, first of all the components that are required for the ubiquitination cascade. I'm sure you know that the first step is ubiquitin activating enzyme, the E1 enzyme. The second step is a conjugating enzyme. In this case, it's UBC7. It's called also E2 enzymes. So you probably hear talks here from Avram Hershko and Aaron Chikanova. They are the ones who gave these names to E1, E2, and E3 because they alluded in different positions on their columns. And they got the Nobel Prize for discovering these ubiquitin-like cascades here. In our case, in the case of UBC7, you need an activating enzyme. It's called Q1. And then we need the ubiquitin ligase. And then we need the ADPase. So the ADPase actually consists of the ADPase itself and a cofactor that consists of two polypeptides, UFD1 and MPF4, which I will be abbreviating by UN complex. And then finally, you also need to purify the substrate. We're using a classical substrate. This is carboxypeptidase Y star or CPY star that has a point mutant in the polypeptide chain, and that single mutation renders the polypeptide chain unfolded or misfolded, and so therefore becomes a substrate for ERAD. So this slide summarizes several years of work by Alex Stein, a former postdoc in the lab, where he purified all these different proteins. Sometimes you see more than one band, but this means that there's a complex of two polypeptide chains. And he also purified the substrate and labeled it with a fluorescent dye so that we can follow it more easily in our assays. So I'm not going to go through all these bands here. In fact, I'll also skip most of the data here. I just want to mention that we found that if you incubate HERD1 with the CPY star fluorescently labeled, you can see binding. But it doesn't, HERD1 does not bind to folded proteins such as the Y-type CPY or these proteins here. Everything is done in detergent. And also, without showing you the data, we find that the substrate binds to the membrane-embedded region of HERD1, which is expected because that's the only thing that the substrate can see from the luminal side. And then we can do the ubiquitination reaction. So what we do is we take the substrate, fluorescently labeled, CPY star, mix it with HERD1, both in detergent, and then add all the other goodies that you need for the ubiquitination reaction. E1, E2, Q1, the activator for UBC7, ADP, ubiquitin. And what you see over time is that the substrate, this is the unmodified substrate, and you get the smear of polyubiquitinated species appearing over time. And this shows an experiment where we left out one of the components at a time, and you can see that the reaction doesn't work unless the Q1, if you leave the activator out, you get a little bit of ubiquitination less efficiently, but in the other cases, completely dead. So surprisingly, what we found is that HERD1 itself also gets ubiquitinated, and actually it's even more efficiently ubiquitinated. So there's auto-ubiquitination of HERD1 happening in the same system. And this was all in detergent, but we can also take the complex of HERD1 and CPY star and reconstitute it into liposomes. So these are artificial vesicles. So basically what you do is you take the complex of HERD1 and CPY star, mix lipid, and then remove the detergent. And so you form spontaneously vesicles that contain the complex. And if you do the ubiquitination reaction, you see again that with CPY star we get ubiquitination. With the Y type we don't, because the Y type protein doesn't bind to HERD1. Yes?
Why do we see a smear instead of ladder?
This is very often the case, and it's presumably because there's not just one lysine that gets ubiquitinated, and so you get sometimes monoubiquitination, sometimes you get polyubiquitination, so it doesn't always look like a nice letter. It's very often the case in ubiquitination reactions.
Presumably, if you only added certain of these components after you send the liposome, you see half of the molecules getting ubiquitinated.
If you only, yeah, oh, you mean because of the sidedness? Yes, so I'll come back to this point actually in a few seconds. So, without showing you the data, it turns out that there's some loop of the substrate that is already on the outside. So if you take the complex of HERD1 and CPY star and reconstitute it, and you try protease protection experiments, you find that some segment of the substrate is already exposed on the outside. So this basically made it impossible for us to use the CPY star as a substrate to actually look at retrotranslocation, because already something was on the outside. And what we really want is to look, you start with something that is completely inside and then see that it moves out. And that would be retrotranslocation. So, this is what we really want. So we want an integral membrane protein, because that's actually much more efficiently integrated into the liposome. If you try to incorporate something into the lumen, it's not very efficient because the internal volume is very small. And so you get only like less than 1% usually into the internal volume. But with integral membrane proteins, you get 100% incorporated, because the hydrophobic segment doesn't like to be in aqueous solution. So you get, of course, both orientations. Sometimes the luminal domain will be, this large domain will be inside, and other times it will be outside. And so what we really want is to look at these molecules and ask whether this domain can move outside. And this would be retrotranslocation. Okay, so that's the goal of the experiment. So Ryan Baldridge, postdoc in the lab, was able to design an assay that would allow us to use to look at retrotranslocation. What he did is he took this membrane protein, CPY star, with a transmembrane segment, and he labeled at the C-terminus with a fluorescent dye by an enzyme called sortase. So you can take a little peptide, label it with fluorescent dye, and add it to this protein. There's to be a recognition signal, and then you exchange the C-terminus with this fluorescent peptide. It's a transamidation reaction. And so you get a fluorescent label exclusively at the C-terminus. So if you now reconstitute this thing into liposomes, so you take mix it with lipid and take the detergent away, you get both orientations, as I was saying before. And then we incorporate afterwards the HERD1 molecule with a procedure that's called directed insertion. So you add just a little bit of detergent, and so the HERD1 molecule will incorporate into the liposomes with a large cytoplasmic domain staying on the outside, because the liposomes are not completely solubilized. So the membrane becomes a little flexible, but it is not enough flexible to allow a large domain to go across. So you get the HERD1 molecules all incorporated with a ring finger on the outside, but the substrate has both orientations. And then there's a big trick here that he invented. You do another sortase reaction now. And So every molecule that has a C terminus on the outside will switch color, so from green to red, whereas all the molecules that have the opposite orientation will stay green. And now we want to look only at the red molecules because they have the large domain inside, and ask whether the large domain can come out. But before we do that, we need to make sure that we actually have the orientation that we claim. So here we're using a modification reaction that will modify all cysteines with a reagent that adds 5 kilodalton for each cysteine that is modified. So all the cysteines are on this large domain, and so as you expect, the green molecules should be modified regardless of whether you have detergent present. So this is the unmodified, this is the modified. But the red molecules, they should have the cysteines protected by the bilayer, and indeed we see no modification unless we add detergent. So this proves that the orientation is correct. We have another way of testing the orientation using a ubiquitin ligase called RSB5. Again, all the modification sites are in this large domain. And again, the green molecules are modified regardless of their of whether you have detergent present or not, whereas the red molecules are only modified when you have detergent present. And here's the important experiment. If you take these liposomes and just add the ubiquitination machinery, you find that both the green and the red molecules are modified. And so this means that the red molecules, which were previously had their large domain on the luminous side, must have moved around across the membrane to be able to receive the ubiquitin. And so this is shown here schematically. So here the red molecules, they started out with a large luminal domain on the inside, and then at least one loop had to move across the membrane to be modified by the ubiquitin ligase. The green molecules had their large domain on the outside right away, and so they could be modified right from the start.
How do you know that it's not the hard one that
Okay. We know from protease protection that essentially all molecules have the ubiquitination site, the ring finger on the outside, almost 100%. Now, this is an important experiment. So if this is translocation, you would expect that the HERD1 molecule forms a channel. And so what we did is we removed several of the transmembrane segments of HERD1. So we made a protein that we call mini HERD1. And you expect that the channel is destroyed by this. And indeed, this mini HERD1 retains ubiquitination activity, but in this case, the red molecules are not modified by this mini HERD1. So in other words, with mini HERD1, this large domain cannot move across the membrane, which provides support for this idea. So the hypothesis based on these experiments is that the HERD1 molecule allows retrotranslocation of a substrate domain, and we postulate that autoubiquitination of HERD1 is required for this process. In other words, the HERD1 molecules self-ubiquitinate themselves, and this allows the large domain to move across. So Ryan actually developed another assay to test for retrotranslocation. So what he did is he modified the large domain with another dye, and that dye we have an antibody to that quenches the dye. So all molecules that have the large domain on the outside will be reacting with the antibody and will be quenched right away. But the large domains on the inside will not be touched by the antibody unless they move across the membrane and become accessible to the antibody. And this is shown here. So when you do the polyubiquitination reaction, you see that this domain moves out of to the outside and becomes increasingly accessible for the quenching reaction. This is just a control showing that if you add Triton, detergent, at the beginning, you get right away quenching. If you don't have the ubiquitination reaction, we see very little quenching over time. Without showing you the data, a catalytically inactive HERD1 mutant doesn't do it. and a lysine-free substrate can also be retrotranslocated by HERD1, supporting the idea that you don't need ubiquitination of the substrate, you need ubiquitination of the HERD1. Now you might ask me, where is the ubiquitination happening in HERD1? And it turns out it turns out to be rather complicated. So the HERD1 molecule, it turns out, is autoubiquitinated at many different sites. So initially we tried to mutate away all lysine residues in the HERD1 molecule. There's about 20 or so. So it was a lot of mutagenesis actually. In the end, it turned out the protein was completely dead. So we had to revert to another strategy. We divided the protein into 3 domains: the transmembrane segment, the ring finger domain, and there's a C-terminal domain, and then mutated in each domain the lysine to arginines. So we have mutants in which only those lysines are mutated, another mutant in which you have all the lysines here mutated, and another one in which we have them all here mutated. And these mutants were at least, the protein was still behaving okay. And so it turns out that the ubiquitination, the autoubiquitination, happens in the ring domain, or I should say autoubiquitination in the ring domain is the one that is important for retrotranslocation. So in this experiment, you can see that if you have a mutant in which the ring finger domain cannot be ubiquitinated, you need lysins, so it doesn't work with arginines. In this case, we don't see the ubiquitination reaction going with this mutant. And also in this other assay that I mentioned with the quenching of the antibody, you don't see this quenching going on with a mutant that cannot be ubiquitinated in the ring finger. So it seems that the ring finger autoubiquitination is required to open the channel to allow retrotranslocation. And in vivo, we see a similar thing here. What you do here is you looking at CPY degradation in vivo by cyclohexamide chase experiments. So what you do is at time point 0, you add cyclohexamide to block biosynthesis of the new synthesis of a protein. Every protein that was made before will then be degraded with time. And so what you can see here is in Y-type cells, you get rapid degradation. But if you have this mutant where the ring finger is blocked from ubiquitination, it's just as good as if you didn't have any HERD1, any lysine present or the empty vector. And we have similar results with 2 other substrates here. So again, this mutant is basically dead. So this leads me to this model here. So we have the HERD1 molecule, we have the substrate. We think the substrate, so then the first thing happens is the polyubiquitination, autoubiquitination of the HERD1 molecule. This allows the opening of the channel, and the substrate, we think, I have to say this is very speculative at this point, can slide back and forth in the channel. When a lysine arrives on the cytoplasmic side, it can be modified by the HERD1 molecule as well. Once you have a polyubiquitin chain on the substrate, it's so bulky that presumably it prevents the backsliding of the chain into the lumen. So it's again a ratcheting mechanism that would allow the substrate to move into the cytosol. But the major conclusion that we think we can draw from this data is that the HERD1 forms the long sought after retrotranslocation channel. There's been a long history about finding the channel for retrotranslocation, and we think that we finally have the right thing. We also speculate that there are other multi-spanning ring finger ligases in ER that function in a similar way, and there are quite a number of them. So here's a list of ligases in yeast and in mammals. I mentioned already the HERD1, the DOE10 is also multi-spanning. And then there's this new complex that Pedro has found. All of them have multiple transmembrane segments. In mammals, there are even more. Some of them are involved in this MHC class 1 degradation that I mentioned. So the assumption is that in all cases, there are polyubiquitination gated protein conducting channel.
All going to the same one.
That's the idea. They all would be gated by autoubiquitination that would open the channel and allow the sliding of a chain.
Are there any dots that are involved in the deubiquitination and thus closing the
Excellent question. This is what Ryan is working on right now. So you would expect that there is a deubiquitinating enzyme that cuts back the ubiquitin chains from HER1 to reset the protein for the next round of translocation. We know there is a different DOP that is required for the substrate deubiquitination than for the HER1 deubiquitination. Now I have one more point that I would like to make, and then we can have more discussion about this. So everything I told you so far was done with this basic ERADL system, and obviously we've been ignoring all the other components. So let me just say that we still don't really understand what these other components, I mentioned this is 5 different proteins forming a complex, and one of the questions is what are they actually doing, and specifically. One thing we do believe is that this HER3 protein is kind of a break. So we know that if you delete the HER3, then the HER1 gets autoubiquitinated very rapidly and degraded. And so we think this protein prevents the autoubiquitination until substrate comes, and then the substrate, there's some evidence that substrate binds to the HER3 protein, and this may relieve the break so that you get autoubiquitination of HER1. So this is the only thing where we have a good idea. The DUR1 is in particular a really mysterious protein at this point. It's multi-spanning, and we really don't understand what it does.
What about
Q1 is only involved in the activation of the UBC7. There's actually a structure on this, so that's pretty understood how it works. Yeah, so that part is.
So it's not involved in, I suppose Q1 is the one that has a Q domain and ubiquitin binding domain.
Sorry?
It has a ubiquitin binding domain, Q1.
Yes, it does.
It's not involved in binding substrate or exactly.
We don't think so, although, yeah, we haven't really looked at Q1 very well in detail. There's a structure of Q1 bound to UBC7. So I don't know much about that actually. So at this point we think that all the linkages are K48. There is no branching of polyubiquitin chains that we think is involved. So let me just briefly talk about, I don't have the time, but just mention briefly the membrane extraction by CDC48. So this is the last step. So it turns out you get polyubiquitination. So the retrotranslocation has happened in the absence of CDC48. So you just need the HER1 protein to get a domain to the other side. But it's not sufficient to extract the protein. So in order to extract it from the membrane, you need the CDC48 complex. And just to show you one slide where we can reproduce this with our in vitro system. This is again Alex Stein's work. So what he did is he took the complex of HER1 and CPY star, we constituted it into the liposomes, then did the ubiquitination reaction, and then put it onto a nicodens gradient at the bottom of the gradient, and then centrifuged. And if you don't have CDC48, what you expect is that the vesicles float in the gradient and end up on the top of the gradient. But if there's membrane extraction by CDC48, what you expect is that the extracted material should stay at the bottom of the gradient and the vesicles should float. And so you can ask whether CDC48 gives you some stuff that stays on the bottom. And this is in fact the case. So in the absence of CDC48 complex, you don't see much at the bottom of the gradient. I should say we're looking here at the membrane extraction of both CPY star and HER1. Remember I told you that in basic ERAD-L, the HERD1 protein is also ubiquitinated and also degraded, like the substrate. So this is why we looking at both CPY star and HERD1. And in the presence of CDC48 complex, you see some materials, ubiquitinated material, staying at the bottom of the gradient. And this is what we call the extracted material. If you quantitate, it turns out there's about 25 to 30% in best cases extracted from the membrane. And here are some controls that show that you need the whole complex of CDC48 complex. It doesn't work without the cofactor. It doesn't work without ADP. So in other words, it seems to be real. So this allows us then to add another step here. So we were, at this point, we had this ratcheting mechanism where the polyubiquitination of the substrate prevented a loop from moving backwards. And now the CDC48 complex would bind to the polyubiquitin chain and pull it out of the membrane. Now you will ask me, how does it really work? And unfortunately, I don't have the time, but actually I have a student in the lab who has actually done a really remarkable work on understanding how the CDC48 complex works. And I'm just going to summarize in 2 slides what he did, what he found. So just to remind you, CDC48 P97 is an ATPase. It has an N-terminal domain called the N domain, and then 2 ATPase domains in tandem called D1 and D2, and each of them have these characteristic Walker A and B motifs that are required for ATP binding and hydrolysis. And as before, it always, it always works with a cofactor, but very often it works with this cofactor, UFD1 MPR4 complex, the UN complex. So 6 of those subunits, 6 of these molecules that I just described, form a hexameric ring structure. So you have the N domain here, the D1 domain is in green, and the D2 domain in red. This is a cryo-EM structure, as also X-ray structures. And then if you look down, you can see there's a hole in the middle and the N domain sitting on the side. And the cofactor binds to the N domain. And what Nick Bodner in my lab has now shown is that the substrate is actually binding to the N domain through the ubiquitin chain and is moved through the central pore. So this is just a summary of the whole thing that he did, and I could show you 30 slides on this, but I'm not going to do that. So here's the substrate. It slides back and forth in the HERD1 channel and gets polyubiquitinated. This polyubiquitin chain binds to the UN complex. It's exclusively through the UN complex. There's no interaction with the N domain. Then the substrate inserts as a loop into the CDC48, into the central pore. And then the D2 ring of the ATPase is the one that reels the polypeptide chain through the central pore. It's doing all the work during the translocation mechanism. In order to release a substrate, you need to cut back this ubiquitin chain. And this is done by a deubiquitinating enzyme called O21. So that's a different DUP from the one that cuts back the ubiquitin chain on the HERD1 molecule. And in fact, O21 cannot work as long as the substrate is not released from the membrane, because there's another component here in the membrane that competes with O21. So as long as the substrate is still not released, O21 cannot do anything. Once it's released from the membrane, the whole complex, then the O21 starts cutting back the ubiquitin chain. Once the ubiquitin chain is short enough, then it will be pulled through the membrane, through the CDC48 complex. Now the surprising thing for me was that Nick found that you can pull an oligo-ubiquitin chain through the central pore. The ubiquitins are unfolded. There's about 5 to 10 that you can pull through the central pore, and then they refold on the other side, presumably. And this allows then the polyubiquitin. So the ubiquitin chain is still long enough so that the proteasome can deal with it. So you need about 4 ubiquitin chains, 4 ubiquitin moieties to allow the recognition by the proteasome. And it seems that it's long enough when it comes out to allow the degradation by the proteasome. And with that, I'm going to stop and just acknowledge the people who have done the work. So Alex Stein, he's now a group leader, young group leader at MP at the Max Planck Institute in Göttingen. Ryan Baldridge is current postdoc, but he's on the job market. And Nick Bodner is an MD PhD student, and I didn't have really a chance to do justice to his work. Thanks.
So regarding HARD1 and the 4, so you draw it as the self-ubiquitination is actually an intermolecular event in which 2 HARD1 molecules have to interact. Is that, I mean, do you have proof that that's
Yes, so the HERD1, HER3 structure that I mentioned, this cryo-EM structure, shows 2 HERD1 molecules and 2 HER3 molecules in the complex. But I have to say we're not sure whether the autoubiquitination is across 2 molecules or whether it's intramolecular. That's unclear.
CDC48 has to be recruited every time a protein goes by, right?
Right.
So how does it know that the protein is coming?
So the most important interaction is through the ubiquitin chain. The substrate is ubiquitinated, will bind the UN complex, and all the UN complex is bound to CDC48. So that's the major. But there is another component called UBX2 that helps the recruitment as well. That's the competitor for O21 as well.
So it's HERD1, it started once we ubiquitinated, it started from the membrane in the same way with P97.
Yes, it looks like. So in this artificial system where you don't have all the other components, the HERD1 molecule seems to be extracted in just the same way. In vivo, in a wild-type cell, HERD1 is actually quite stable. So it's not normally ubiquitinated and extracted. It also wouldn't make much sense to degrade the channel every time you move up. No, it's because of the other components that are present. So HERD3 is a major break to prevent that, but presumably the other membrane proteins are also keeping the protein in the membrane, so preventing its extraction. This is a good question still. We need to get a system where we have all the components present in our reconstituted system. And we have been able to do that, but there are still issues with the system. So So if I understand correctly, so we are postulating HERD1 is doing all the ubiquitination of the substrate in this ERAD-L pathway. There are no other ubiquitin ligases required for this. But there is a good question here of how do you transfer the substrate from CDC48 to the proteasome. There are, Bob Sauer has postulated that the CDC48 complex would associate directly with the 20S proteasome. That may be true for some substrates, but we don't think it's a general mechanism. So we believe that you actually remove the substrate from the trans side of the CDC48 complex, and then they're transferring it with so-called shuttling factors to the proteasome.
So do you know you still need the ubiquitins to be there? Because that would be the recognition by the proteasome.
Yeah, well, we think so, because ERAD requires these shuttling factors, RAD23 and DSK2, and so based on that, you would say you need the ubiquitins because they recognize ubiquitin.
What was the in vivo trigger for the polyubiquitination of HERD1?
We think the substrate binding then to the HER3 protein will relieve the break that will allow polyubiquitination.
Have you tested the hypothesis that the polyubiquitination on first one is not to open up the channel, but for the catalysis? Have you
For catalysis.
polyubiquitination?
Right, so it's presumably both, actually. So you for
If you reconstitute without the polyliposome, but just in a homogenized lysis, do you still have the ubiquitination reaction going on if you don't have the polyubiquitination of the first one?
you don't. So so it's the same thing. So these ring finger mutants are defective in doing the reaction. Yes. Yeah.
I have a very best question.
Although the ubiquitination still works, the retrotranslocation doesn't work. So these ring finger mutants that I mentioned, the lysine mutations, are active in ubiquitination, but they're not active in opening the channel. So you need the ubiquitination on the ring finger in order to open it. It's not the activity per se.
I was wondering if there is a proteasome-mediated degradation that happens inside the lumen, the ER, and not.
No. Not.
And that's why
There's no proteasomes inside the lumen of the ER. So they're all either in the cytosol or in the nucleus.